Hello everyone. Today I am going to introduce you about 10 basic terms which are most commonly used in the subject microbiology. So let's start. The first term I am going to introduce you here is culture. So as you can see here on this slide, I have included two pics. In first pic you can see growth of microorganisms in the form of microbial colonies. And in second picture you can see growth of microorganisms in terms of turbidity in test tubes. The growth of microorganisms, whether it is on the solid, any kind of solid medium, solid base, solid support as in petrificate, or whether it is in the liquid medium as shown in the test tube, is called as culture. So we most commonly use this term in microbiology laboratory, in microbiology subject, like this culture is of which bacteria? It is of E. coli. Store this culture in the refrigerator, take this culture, bring this culture. So it means what? The culture is what? Microbial growth. Now we are going towards second term. Second term which we should know about is pure culture and consortium. So what is pure culture? As you can see here in the pic A, I have shown that any kind of solution, if it is having same type of microbial cells, like I have made here cocci, circular in shape, round shaped, spherical we can say. So then this type of culture will be of pure culture type. Means it is having single type of microorganisms, microbial cells. Now we are going towards second type, B. B picture is going to show us that if any kind of solution, different type of microbial cells are present, different kinds are there, like you can see here, cocci, spirali and rod shaped. And of course, in next picture you can see here corresponding to it different colored colonies. It means that it is having mixed type of culture. So mixed type of culture, for this in microbiology we use the term consortium. Now let's talk about the third term. It is media or medium. So microbiological medium, media, these kind of terms we use to represent what? Nutrient solution, what we use in the microbiology laboratory to support microbial growth. Now fourth term, broth, agar. These are also another kind of terms which are used in microbiology subject. Broth generally refers to that kind of medium which is liquid in nature. Like in A picture you can see here, liquid medium is there. If nutrient solution is liquid in nature, medium is liquid, then we use the term broth. And if the medium that is going to support the growth of microorganisms we have taken in the laboratory is solid, then we are using the term agar. Why agar? Because in B picture you can see petri plate is there and solid medium is there. Because same composition we can take of any medium as we have taken for liquid medium, in that we will be adding a solidifying agent which is known as agar. That's why we are saying that is agar medium. So broth is the term for liquid medium and agar is the term for solid medium. The next term I am going to introduce you here is about sterilization. It is very important in microbiology. Everything whatever we use in microbiology laboratory for experimental purpose should be of sterile nature, and it needs a process to become sterile, that is known as sterilization. As it is clear from the picture, if you have taken any kind of flask, you have prepared nutrient solution or medium for microorganisms and you are interested to isolate microorganisms from food sample or say water sample. Maybe that flask is already bearing some microorganisms, or that solution, water, nutrient ingredients, they are already having some associated microbes. So they can contaminate, they can interfere with your results. You are presuming that I have got this result from the milk sample, but maybe those kind of microbes were already present on the surface of the flask. So sterilization is very important. In sterilization we completely remove all living objects, entities from what? From any kind of material. It can be glassware, it can be medium. So picture is showing where microbes are present, that is non-sterilized one, and where it is completely free of microbes, then we are saying now it is sterilized. For sterilization we use autoclave, hot air oven, such kind of instruments are used in the microbiology laboratory. So another important term which is used in microbiology is inoculation. Inoculation is actually the term which we use in microbiology for the transfer of any kind of material, any kind of culture, any kind of sample into the medium. Like you can see here, I have shown a picture of soil sample. So if I am going to transfer soil sample into nutrient medium so that nutrient medium can support the growth of microorganisms present in the soil sample, then this type of transfer is known as inoculation. Means I am going to inoculate soil sample into the medium. In the same way, when I am going to take a stored culture and I am going to revive it, means I am going to reactivate it, again then I will be using the same term, inoculation. Inoculation of old culture into fresh medium. So next important term is incubation, which everyone should know as a beginner. Incubation is actually the term which is used to for the time period which is required for the microorganisms to show us a kind of growth which is visible to our eyes. Like if we have used any kind of sample, say water sample, and we have applied water sample on microbiological media, so immediately we cannot get results in the form of visible colonies. What is the number of microorganisms in the water per ml of the water sample? It needs some time. So that time span which is required by a particular microorganisms to form colonies to give us a visible growth, that time period is known as incubation in microbiology. The next important term is isolation. Isolation is very commonly used mainly when we are going to study microbial population of a particular sample. Like in laboratory we have received soil sample, any pharmaceutical product, any food product, that we need to study for its microbial population, that how many bacteria are present per gram or per ml of this sample. Then we will be following a process. That process of studying microbial population from a sample is known as isolation. Next important term what every beginner in microbiology subject should be known about is contamination. Contamination is a very serious concern in microbiology subject. Why? Because contamination is actually the term which is used when some undesirable microorganisms are present in a tube, in a sample where we have actually stored only one type of microorganism. Like you can see in picture A, I have shown a stored culture which is present in the vial, means growth of microorganisms what I have stored in vial number A. It was actually of cocci, circular shape bacteria, and in picture B another tube is present having same culture. But when I am going to revive this culture, I am going to activate this culture. When I am going to take it out from the refrigeration, what I observe in A picture, A tube, what happened? Circular shape bacteria means cocci appeared. On the other hand, in B picture, what I have used tube number B, what I have observed, different type of bacteria were present. Along with cocci, spirali were also present, rods were also present, means some undesirable microorganisms or microbial cells have entered into that tube. Maybe that tube when I was going to store my cocci culture was contaminated at that time. So what it has done, the tube or any non-sterile conditions what were prevailing over there at that time, they have contaminated this culture, which is now not of our use. So contamination is a serious problem. Contamination, again we can learn about the growth of undesirable microorganisms in culture of our choice. So this is the last term what I am going to introduce you here is about subculture or subculturing. Subculture is very important term which is generally used when we are going to make a second transfer of already grown culture. Why the second transfer we are going to make? To purify the colony or to purify a particular bacterial culture. Like you can see here in picture A, I have shown many colonies are growing. And what we will be doing in order to purify them, we will pick a single colony and then we can apply it on a fresh medium plate. So that now only this colony will be further multiplying and same type of microbes or pure culture we can get here. In the pick B, suppose in first plate we got two type of or mixed type of culture. When we are going to transfer a little amount of culture on fresh medium plates, what will be happening? Only that single colony will be growing on the another plate. So in this way our culture will be purified. Subculturing we can say now is a mean of purifying the culture, which is used in the laboratory, or we can say second transfer of already grown culture, then we use this term subculture. So this was all about the basic terms, what I have introduced you here, and I hope they will surely helpful or useful to microbiology beginners. Stay connected through my video clips. Thank you so much for watching.